I'm like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my
Welcome to the National University of Singapore, Yong Lu Lin School of Medicine's Healthy Longevity webinar series. Once in a while I get that right, actually. Thanks for coming. As you know, some of you who've been watching, I've been traveling in various parts of the world. Well, I'm back in Singapore now, and the first thing I can report is this VTL vaccinated travel lane is the best thing ever. I was able to get through the airport, get tested in 20 minutes, and just had to wait for my test result, which was negative. And within a few hours, I was released into the wilds of Singapore. So this is much better than 2-week quarantine. Any of you thinking about that? We're lucky tonight. We have Adam Antebi on talking about his research on aging, and we'll get to that in a little bit. I'm sure you're going to have questions, so please use the Q&A function to ask any questions you have. And Dong Lu, our moderator, will be able to take those questions and ask them to Adam. Before we do that, though, we're going to have Katrina Lamalu on, an internship student in my laboratory, and she'll be talking a little bit about her project on glycine's effect on the microbiome and health span in mice.
Hi, thank you for the introduction. I'm Katrina, and I'm a Polytechnic intern at Brian Kennedy Lab. And I'll be sharing about my final year project, Effects of Glycine on Microbiome and Health Span of Aging Mice. So glycine is a non-essential amino acid which is synthesized and produced in the body. Generally, amino acids which can be produced by the body are considered non-essential, while amino acids which cannot be produced by the body, such as threonine and valine, must be consumed and are therefore considered essential. While our bodies can make glycine, it can also be found in many high-protein foods like red meat, chicken, dairy, soybeans, granola, and eggs. Glycine performs many functions in the body and is essential in the production of important substances like proteins, collagen, the antioxidant glutathione, among others. Substances like collagen and glutathione serve beneficial functions but are also produced by the body less with age, and so supplementing glycine into daily diets may be beneficial for health and aging. In this study, the researchers looked at mitochondrial respiration of fibroblast cells, which are cells found in connective tissue, from young and elderly human donors. They are depicted in the black and white bars respectively. Here on the graph on the left, they show that elderly fibroblast cells show age-associated mitochondrial respiration defects with lower oxygen consumption rates compared to the young fibroblasts. They then treated the elderly fibroblasts with glycine, and glycine was able to reverse the defects to almost the level of young fibroblasts, as seen by the white bars of the graph on the right. And so, the author suggests that glycine supplementation could prevent age-associated respiration defects and benefit healthy aging. What this means is that having glycine can improve mitochondrial function, which is important for cells to generate energy to work properly. In terms of glycine and aging, a few studies have assessed glycine's ability to extend the lifespans in animal models. Glycine was supplemented to mice in one study and to C. elegans worms in another, and they found that glycine was able to extend the lifespan of mice by 3% and the C. elegans by 33.3%. While the improvement in lifespan in rodents seems modest, the health span during the study period is unknown. However, previous studies have assessed the effects of glycine on aging and disease models. An example of this is giving glycine to inflammatory bowel disease mice models, and they found that the treated mice have better gut microbiota profiles. In this study, as seen in the first two graphs, glycine fed to mouse colitis models, mouse with colon ulcers, was shown to reduce interleukin-1 beta levels, which contribute to further inflammation, and increase interleukin-10 levels, which is anti-inflammatory and reduces agitation of the colon. The authors found that interleukin-10 expression increased with higher abundance of bacteria Clostridia and Clostridials, and interleukin-1 beta expression decreased with an increase in Ruminococcaceae bacteria. This shows that on top of changing inflammatory profile of the diseased mice, glycine also caused changes to the gut microbiota. From the rightmost graph, glycine was able to reverse the effects of colitis on the gut microbiota to similar to that of the controls. And since glycine has been proven to have a beneficial effect on human and mice health, as well as the gut microbiota, I was interested in the effects that glycine would have on the microbiota of aging mice. So, over the course of my internship, I'll be looking at old mice that we fed a high and low dose of glycine, and I'll periodically assist in assessing the safety of the mice, as well as collecting fecal samples from the mice and extracting the DNA of the bacteria in the fecal samples to look at the gut microbiota. And I hope to see a different doses of glycine affect the gut microbiota of aging mice, and to see if glycine affects the health and well-being of aging mice differently between their sexes, as most studies have either been done on only male mice or disease models. Thank you for your attention.
Thanks a lot, Katrina. We're excited to hear the outcome of that research. It's great to have Adam Antebi on tonight. I've known Adam for a number of years now, and he's always been doing cutting-edge research in the aging field. He got his PhD originally, however, from working on yeast cells from MIT. I am always skeptical of people that get their PhDs from MIT, but actually Adam's really good, so hang in there. He then went to do a postdoc at Johns Hopkins before becoming an independent group leader at Max Planck Institute in Berlin, and then moving back to the US, where he was a professor at Baylor College of Medicine. In 2008, he went back to Germany and joined the Max Planck Institute for the Biology of Aging in Cologne. I was lucky enough to visit Adam in Cologne last week and hear about some of the exciting stuff. So I got a preview of what he's going to talk about today. His title of his talk is Convergent Mechanisms of Longevity. Adam, it's great to have you on the show.
It's great to be here, Brian. Thanks so much for having me. I'm really excited to give this seminar, and I hope everyone there is doing well. Let's see if I can boot this up now. So yeah, again, thanks, Brian. It's really great to be here. And thank you for allowing me to share my work. So I think aging is something that we all can appreciate, and some of us actually are experiencing as we speak. And I think, unfortunately, it entails a decline in health and vitality with time. And it's really vexed humankind since antiquity, because it's only a few steps away from our own mortality. Yet, what can we do about it? And I think this is really what we would like to understand. How can we age gracefully and with good health? And I would argue that some of the greatest insights into the process of longevity and protective mechanisms that stave off the ravages of aging have come from insights from model genetics systems, such as worms, yeast, flies, and mice, as well as fish and other model systems. And the reason they're so important is they allow us to establish causality. We can do interventions, genetic or otherwise, and see the impact on health and even the lifespan. And many of these pathways and interventions are evolutionarily conserved and can inform human aging. Now, the worm is the model system that we use, and it's my favorite model system because it's incredibly powerful as a genetic model, and it only lives a month. And this actually led to the discovery of the first single gene mutants that prolong animal lifespan by Michael Klass back in the 70s and 80s. He performed genetic screens to look for to look for mutations that could extend the lifespan. And one of these mutants that he found was called AGE1 and followed up by Tom Johnson in the 80s. And this mutant lives 80% longer than wild type. So imagine that you could live 150 years. Now, in a parallel line of research, Don Riddle and colleagues were studying a long-lived stage of the worm called the Dhfr. And these Dhfr animals can live 6 months. They go into a hibernation mode, and all these protective mechanisms are turned on during that time. And then when times get good again, they come out of it and they reproduce as adults. And Cynthia Kenyon, when she was at UCSF, decided to study these so-called Dhfr formation or DAF genes. And one of these genes, called DAF2, remarkably doubled or tripled the lifespan of the worm. So instead of living 20 days, it lived close to 60 days. And furthermore, she could show that this longevity was dependent upon another DAF gene called DAF16. When this gene was obliterated, the lifespan extension was gone. And this gave rise to a very profound idea that longevity was actually regulated through genes. Now, the molecular identity of this pathway turned out to be the insulin IGF signaling pathway. Notably, DAF2 is an insulin receptor, AGE1 is a PI3 kinase, and DAF16 a FOXO transcription factor within this pathway. And a modest reduction of this pathway could extend the lifespan through improving health and stress resistance. Quite remarkably, then this explains how a single gene could have such profound effects through endocrine control, through a hormonal mechanism. And this was shown to be the case in fruit flies and in mice and perhaps even in humans. And this, I think, was the big surprise, that this conserved pathway could have similar impact on lifespan throughout taxa. It really cements the idea that comparative biology of aging is very instructive, that there's a conservation of these protective mechanisms that we can learn from. And so since that time, we now know that there are several other conserved metabolic signaling pathways that can regulate lifespan in various model systems, including a reduced level of mTOR signaling. So mTOR is a nutrient sensing pathway, which is responsive to nutrients and growth signals. And when you dial that down a little bit, you extend the lifespan in many different species. Also, removal of the germline stem cells in worms and other species evidently promotes longevity. Diet restriction or intermittent fasting, many of you may be familiar with as a health promoting intervention. And in C. elegans, this also is true. You can extend lifespan by about 20%. And then finally, reduced mitochondrial function also has a surprising impact on extending the lifespan. Now, these are some of the major canonical pathways, and people have analyzed these in a piecemeal fashion and found, for example, specific transcription factors which mediate the longevity effect. If you remove these factors, you lose the longevity. And this pathway specificity has been very important. We've contributed to this knowledge. But in recent years, my lab has become enamored by, I think, a more profound question, and that is, are there convergent mechanisms? Are there convergent regulatory tiers or convergent processes or modules that all these pathways impinge on? And we think this is important because then we can get at the heart of longevity. And in answer to this question, we've identified a number of different levels of regulation. For example, years ago, we and others showed that there's a number of helix loop helix transcription factors, namely Mondo A, Max-like, and Dhfr, which work in a tight regulatory network to promote lifespan extension across all these pathways, and mutation of any of these factors abolishes or abbreviates longevity. We also showed that the nucleolus is an important point of regulation in all these pathways, and that small nucleoli are a cellular hallmark of longevity. And this evidently works through setting the rates of ribogenesis, and which is the major function of the nucleolus as an organelle in producing ribosomes. And this impacts the proteostatic load on the cell and the whole translational machinery. today I want to tell you about our studies on metabolic modules that we think are convergent to all these pathways, namely the folate and methionine cycle intermediates. And this is the work of a very talented postdoc in my lab, Andrea Antebi. Andrea is an analytical chemist, and he came to my lab and asked the question, what do the metabolomic profiles look like in these worms? What does the spectrum of metabolites look like in long-lived animals, and do they share anything in common? And so he performed metabolomic profiles on high-resolution mass spectrometry, looking at the various strains that I just showed you. He identified metabolites that were differentially regulated with respect to the wild type, and we could put these into metabolic pathways and then leverage the changes between the different pathways to see what was changed in common. Once we identified those pathways, we could then knock down gene function through genetic tricks like siRNA treatment or metabolite supplementation to see if they were causal for longevity. So that's exactly what approach we took. And this just shows you a heat map of about 150 soluble metabolites that we looked at. And these are the 4 genotypes that we looked at. This is the germline-less GLP-1 notch, the Dhfr insulin receptor, the iron sulfur protein affecting mitochondrial function, and E2 in diet restriction. And blue means downregulated, and red means upregulated. And there are many things that are changing here. We think this is an incredible resource for the field. But our attention was drawn very quickly to folate and methionine metabolism intermediates. And for example, 5-methyl tetrahydrofolic acid was robustly downregulated in these 3 genotypes, upregulated here. And S-adenosyl methionine, a methionine cycle intermediate, was downregulated in all 4 pathways. And I should say that we also use computational methods, and these also very strongly pointed to the folate and methionine cycle intermediates as important. So what is the folate cycle? Well, folate is a vitamin that you get from the diet. It's vitamin B9, and it's taken up and it's used in 1-carbon metabolism in the transfer of methyl groups, formyl groups, or methylene groups to different chemical moieties important to anabolic processes. Namely, it's involved in the production of methionine. It's also involved in the production of thymidylate, which is important for DNA synthesis. It's also important for NADPH and NADP ratios, which are important for various biosynthetic reactions, affects amino acid pools. You heard about glycine earlier. Probably some of the effects of glycine may be through these pathways, and it also affects purine biosynthetic pathways. So it's basically an anabolic pathway when it's upregulated. And so Andrea decided to do targeted metabolomics in this case, where he can zoom in on folate cycle intermediates and measure it in their levels in all these different genotypes. And I think you can see in red, there are many of them that are upregulated, others are downregulated in blue. And in particular, he could recapitulate what he saw with the low levels of 5-methyl tetrahydrofolic acid or 5-methyl THF. And so then he asked, well, which of these enzymes involved in the cycle may impact longevity? And so what he did was he knocked down their activities through RNAi, and none of them had an effect except for dihydrofolate reductase 1 and thymidylate synthase 1. And interestingly, both of these enzymes are rate-limiting steps within the folate cycle, and they actually form a complex. So this just shows you the data. Here is DHFR1 knockdown, TIMS1 knockdown. Black is wild type. We see about a 20% increase in the lifespan. For reference, the insulin receptor gives about a 200% increase. And then we asked, well, when we knock down the DHFR1 enzyme, what is the impact on folate intermediates? And so we did metabolomics again, and we see shown here in orange is DHFR1 knockdown. We accumulate folic acid, but we deplete all the other folate intermediates compared to wild type shown in black. So orange is down except for folic acid itself. And so this raised the question, is longevity arising due to elevated folic acid or decreased folate cycle intermediates? And we could actually test this idea by supplementing with folic acid. And in this case, it had no effect. when we supplemented it to DHFR1 knockdowns, they were still long-lived. But when we supplemented 5-methyl-THF, this metabolite here, we abolished the lifespan extension. And along those lines, just feeding them 5-methyl-THF could replenish the whole folate pool. So this suggests then that low levels of DHFR1 lead to low levels of 5-methyl-THF, which leads to longer life. We then asked, well, is this true for just lifespan or also for health span? And we can challenge worms with proteotoxic aggregate-prone species that compromise their health and see how they perform against that. And this is a model of Huntington's disease, polyglutamine repeat disease. And this leads in wild type to progressive paralysis. They slow down their thrashing rates in adult life with time. But when we knock down DHFR1, that process is slowed down dramatically. We double their motility essentially, but this is dependent on 5-methyl-THF. Feeding them this metabolite reverses the effect. And similarly, this species causes aggregates. Those aggregates are ameliorated by DHFR1 knockdown, but reversed by 5-methyl-THF. So our conclusion is that low levels of this DHFR1 activity are beneficial for both longevity and health span. We then asked, well, what about the other pathways that are starting point in all this? So we had started with insulin signaling and mitochondrial function and seen that levels of this metabolite, 5-methyl-THF, are down. What happens when we supplement with that metabolite? And so here's an insulin receptor mutant, long-lived, but when we give them 5-methyl-THF, we abbreviate the longevity. Similarly, with the ISP1 mitochondrial mutant, we abbreviate the lifespan. And so this suggests that at least part of the longevity of these different strains is due to the regulation of the folate cycle intermediates. Clearly, there's another component that has yet to be discovered. Maybe some of our other metabolites will come to the fore. And importantly, 5-methyl-THF itself to wild type has no effect. So it's not just a general toxicity. It really doesn't reduce the lifespan of a wild type animal. We could also show that this is part of a signaling pathway, since if we look at the messenger RNA of dihydrofolate reductase in an insulin receptor mutant, it actually goes down. So this means it's transcriptionally regulated. And we see the same thing in terms of ISP1. So we have yet to identify how it's regulated, but I think this suggests indeed that we have a signaling pathway whereby, say, low insulin signaling results in low levels of the messenger RNA, low levels of 5 of the activity, and low levels of this metabolite leading to longevity. Now, the question is, what's the mechanism? And here we hypothesize that the mechanism might be working through methionine cycle intermediates, since folate and methionine are quite intimately linked. Notably, 5-methyl-THF donates its methyl group with homocysteine to make methionine through the actions of methionine synthase, or METR1, in the worm. And so we asked, does dihydrofolate reductase knockdown affect methionine cycle intermediates? And indeed, shown in orange, you can see that methionine and S-adenosyl methionine are down relative to wild type, shown in black. And the precursors to this biochemical reaction, homocysteine and adenosyl homocysteine, are up in the DHFR1 knockdown. And all of these phenotypes are reversed simply by feeding them 5-methyl-THF. So evidently, knockdown of DHFR1 leads to methionine and S-adenosyl methionine restriction. If this is true, then knockdown of methionine synthase and DHFR1 should give similar metabolic spectra. And indeed, we see that for methionine, we see that for S-adenosyl methionine, we see that for homocysteine, as well as a number of other metabolites, which are commonly regulated by these two enzymes. And finally, if methionine restriction is causal for longevity, then feeding back methionine to the worm should reverse the phenotypes. And that's exactly what we saw in a dose-dependent manner. Methionine will reverse the longevity, yet it has no effect on wild type. And so we suggest that restoring the methionine pools is abbreviating lifespan, and restricting them is extending. Now, this is very exciting to us because across all these different pathways, it seems that folate and methionine restriction seems to be playing a role, but we weren't the first to show that methionine restriction is causing longer and healthier life. It was already shown back in the 90s in rats, and it was shown in other species later on that methionine restriction confers health benefits similar, but not completely the same as diet restriction. But I think ours is the first to really identify this as a convergent mechanism across different longevity pathways. Now, I've just spent a lot of time trying to convince you that the beneficial effects of DHFR1 knockdown are due to the methionine restriction, but I throw this out there for Brian because the beauty of the metabolome is that we can see lots of metabolites change and look at the interconnectedness and the correlation among them. And for example, we find that when we knock down DHFR1, we see nicotinamide mononucleotide be elevated. Similarly, we see alpha-ketoglutarate goes up. And both of these metabolites, their levels are reversed by feeding them the 5-methyl-THF. Both of these metabolites are gerometabolites. They on their own have been shown by others when supplemented, to extend the lifespan. So it could be that what we're seeing is an interconnectedness of methionine restriction to these metabolites and perhaps mitochondrial biology. So, and then last, we wondered, well, if it's true across pathways, what about across evolution? And here we were very fortunate to work downstairs from my colleague, Linda Partridge, who's studying insulin signaling in mice. IRS knockout mice have low levels of insulin signaling and they're longer lived. And so Andrea did metabolomic profiling comparing the worm insulin signaling mutant DAF2 in IRS mice. And what he saw was that 5-methyl-THF is down in both species, as is S-adenosylmethionine of the methionine cycle. So evidently, both in worms and in mice, reduced insulin signaling reduces levels of folate and methionine cycle intermediates. And so I think this is really exciting because methionine is such an interesting and complex metabolite and it really ramifies into many different aspects of metabolism and growth. And so it raises a question, well, how does this metabolite actually impact longevity? And there's several possible ways that we can imagine. Methionine, of course, is the initiator. Methionine begins protein synthesis, and it could be that by restricting methionine, we slow down this process and increase fidelity. S-adenosylmethionine is used to add methyl groups to many different proteins throughout the cell, including to ribosomal RNA, again, which is important for the processing and of this ribosomal RNA and could impact protein synthesis itself. S-adenosylmethionine is also used to decorate histones and DNA. So the methylation of those components regulate the epigenetic state of the organism. And in particular, what's quite exciting, many of you may have heard about the epigenetic clock, the DNA methylation clock. We can clock chronological and perhaps biological age by looking at DNA methylation marks. And perhaps speculatively, this perturbation of methionine cycle pools is somehow linked to that clock. And we'd love to really explore that idea into the future. It's also been shown that S-adenosylmethionine is regulating the mTOR pathway. And we've also seen that mTOR regulates S-adenosylmethionine. So there's a kind of a magic circle that we think is happening that sets up this kind of state of the organism, metabolic state that is beneficial. And last but not least, homocysteine can be transferred into the transsulfuration pathway, which affects glutathione synthesis, cysteine metabolism, and H2S production. And so these may play an important role as well in regulating mitochondria and lifespan. So then what are some of the translational aspects here? And I think we're still pondering this, but actually there are well-known drugs that inhibit DHFR dihydrofolate reductase in humans. Methotrexate is one, and this is used to treat cancer. So at high concentrations, it causes a disruption of DNA synthesis and triggers cell death in certain cancers. Obviously, that's not beneficial, but at lower doses, it's used as an anti-inflammatory or to treat autoimmune diseases. And we suggest perhaps methotrexate is altering the metabolic state and could have beneficial effects in some circumstances. In fact, there are some meta-analysis that it does have some benefits on heart disease. These pathways, so the folate methionine pathways, are really regulated through the actions of vitamins that we take up from the diet, B9, B12. Many of you are probably taking these vitamins, and we think it could be very important to understand what is the balance for health and longevity. And I think it would be I think it's probably going to be somewhat complex. I don't think restricting these vitamins all the time is going to be beneficial, nor will it be beneficial to have them elevated, but rather coming up with a regimen that is beneficial for both survivorship and fidelity, as well as the growth of the cells. And then finally, I think we can begin to think about different types of diets, perhaps low in methionine or otherwise, that or low sulfur amino acid diets, which could be applied to people to see if it ameliorates age-related disease. So those are some of my thoughts. And then just to summarize again, I've told you that there are multiple longevity pathways in the worm which converge upon folate and methionine cycle intermediates as a convergent mechanism of longevity. And this evidently seems to be true not only in the worm, but at least to some extent in other species like mice. And I think bring this back to the original idea that there are single genes that can extend lifespan. There are single metabolites that can do so as well. And they can extend or shorten lifespan depending on the context. And so we coined the idea of gyrometabolite. And, these may be working through these single gene pathways, such as low insulin signaling and low levels of these intermediates. And finally, we think these metabolomic profiles are really a great way to understand pathway connectivity and allow novel entry points to understand, to improve health. And it's an alternate way of doing so without genetic engineering, let's say. And so, and we think our metabolomic profiles can serve as a great resource to the field to identify other convergent metabolic models of longevity. And so with that, I'd like to thank the people who did the work. So Andrea Anabal, Mary Bell, Hannah Marcus, who are 3 incredible master students, Christian Latza, a great technician, my collaborators, Linda Partridge, Sebastian Grunke, my fantastic and fun lab. They're all out partying. Today's the first day of Carnival, so I'm here alone in the Institute. You can see they know how to have fun. And then thanks to my sources of funding, and thank you for your attention, and I'd be happy to take questions.
Thanks a lot, Adam. And I have to apologize. I didn't mean to take you away from Carnival.
Don't worry. It lasts until February. Today is the first day of it. The first time I came to Cologne, I knew nothing about it. I took the train into the Cologne Central Station on Elfte, the 11th of November at 11 o'clock and 11 after. And I arrived just at that time. And there are all these carnival bands playing in the station. And it was really, I said, wow, this is a great town. I'm really excited.
Oh, no, it was all there for you, I'm sure.
Yeah, exactly. Yeah. I was.
So that's a great talk, and let's start focused and then work our way out to broader aspects of aging. Maybe that would be a good way to go. And I have to say that every time we do an experiment in yeast and even in mammalian cells, we get back to folate cycle, methionine cycle, 1-carbon metabolism. And we sit there and puzzle over what that might mean in terms of aging. And you mentioned a lot of these pathways, but you have methionine restriction, you have methyl donor groups for epigenetics, you have glycine levels, you have homocysteine levels, you have glutathione levels. All of these things have been linked to aging. And there are people that will argue that each of those outputs are important for longevity. do you think it's one or even two of those outputs, or is it a coordinated effort managing all those different outputs?
Yeah, I think it's a great question. My feeling is that it's a combination. That I like to think of the idea of a magic circle of events of regulation at many different levels, and they are coordinated and they're attuned to one another. So it might not even be so easy to separate these different events from one another. I think perhaps with the power of C. elegans genetics, we might be able to isolate different components of this, but it may be challenging. But yeah, I think it's really a lot of pathways seem to converge on this because it's kind of like the metabolic equivalent of mTOR. And in fact, it's connected to mTOR. It's an anabolic pathway that's dealing with amino acid, nucleotide pools, lipid metabolism, reducing equivalents. So it's very central and has a profound effect.
Yeah, I mean, I think that this has really, to my mind, been the challenge of the aging field. We've made a lot of progress in various areas, but I think everybody or most people thought that when we had single genes that could affect longevity, we could work downstream and get to mechanisms. And when we work downstream, we first of all get feedback loops that go back upstream, and second of all, we get changes to everything we look at, and I think it's certainly made me wonder, and like you say, maybe there is this magic circle. I'm not sure why, but it seems like no matter where you intervene, you affect all of these different things. And so,
well, I think that's, that's one of the reasons why we jump from pathway specific mechanisms to really try to understand convergent mechanisms, because it seems like everyone is working in their little niche without even realizing that they may be working on very similar things. And I think it's important to try to bring this together and ask where are the critical nodes that you can intervene to really, in a way that is healthy and beneficial.
Yeah, I have to say the methotrexate idea is great. And actually, Michael Palominas at Texas A&M who works with us on yeast and worms. And also Matt Kaberlein and I, we've been helping get a strategy started to test its effect on mouse aging. So hopefully we'll get some data on that. I think
that's great.
Really intriguing small molecule to look at. And actually it does have toxicity profile, but it may be manageable at lower doses too. So
yeah, I think that's the point. And I think the beauty of it also is there are many people who are taking the drug as a to ward off autoimmune disease. And so we're also trying to get some patient material to see if there's anything to see.
Yeah, and that may be intriguing too, because if you look at things like rapamycin or AKG and others, they seem to be, having effects on autoimmune disease or at least be anti-inflammatory. So that may be another common feature of these longevity interventions.
Yeah, I was really excited to see the AKG effect of dihydrofolate reductase 1 because of your data on that. It's really exciting. And I think, that's a relatively non-toxic way of perhaps modifying these metabolic networks.
Yeah. You mentioned DHFR. There's obviously a lot of work on DHFR. Have there been links to longevity in mouse or human data for DHFR that you know of?
Not really, to my knowledge. I don't think anyone's looked at it. What we're doing is we're knocking down the activity, not knocking it out. Knocking it out would be presumably lethal and would, disrupt cell division and so on and so forth. So, but as I said, people are looking at methotrexate in a as sort of a meta-analysis of methotrexate and its impact on other age-related diseases. And there is some evidence, for example, that it can be beneficial towards heart disease. And what that means, and whether that's completely reproducible, I think remains to be seen. But I think, just these meta-analyses may be able to help shed light on whether these interventions are helpful. I think also the dietary interventions may be really helpful.
Yeah, coming to that, methionine restriction, maybe you can, I'm sure the audience is thinking about this anyway, maybe you can discuss the similarities and differences between methionine or amino acid restriction and traditional calorie restriction, which in animal models is often, or invertebrate models is often carbohydrate restriction in some cases. So, what's the difference between reducing certain macronutrients versus others?
Well, Rich Miller would be better positioned to give you all the details on this. He actually did a very nice comparative study on diet restriction versus methionine restriction. And there are a lot of similarities, but there are also some differences, which, I think, which elude me here. I just don't recall what they are, but, they both share many different benefits.
Yeah, I think with methionine restriction, it's a bit of a challenge to have low amino acid diets. And that's one of the reasons that it's been less work done on it. I know Rich has done some good work and others as well. But it's a little bit harder even in a mouse to do the appropriate levels of methionine restriction. That's one of the reasons we got interested in glycine, because apparently one of the side effects of glycine is that methionine levels are reduced through these pathways. So
yeah, so glycine is it's part of the folate cycle, actually, in I think it's some of the purine biosynthetic pathways. So that's probably where it fits in somehow. So yeah, there's a lot of interconnectivity.
Yeah, the ITP program with Rich and others, they showed that glycine extends lifespan, but it's a small effect. But still, yeah, it's intriguing. And there's some human sort of association data as well. So interesting. I think we're not going to understand aging until we understand the role of these pathways, because I just keep coming back to it, even though I don't always want to. So there's something going on.
I think also what we're excited about is is this the molecular underpinning of the DNA methylation clock?
Yeah.
I mean, it seems kind of like a reasonable hypothesis. And, it could be that, the methylation state of the DNA is influenced by methionine and folate pools with aging. And so, that's a hypothesis that we'd love to test.
Yeah, I think it's a great idea. And it also raises the question, which I still think is not answered, as to whether these methylation changes in DNA that can come up to main the clock, are they just a measure of aging, or are they causally affecting aging? And I don't think we really know yet.
No. Right now, it's a very powerful system as a biomarker, but causality remains to be seen.
Let's take a step back to worms, because we haven't had many worm speakers on yet. And, one thing striking about C. elegans is that you can get such a dramatic extension of lifespan. And if you look at yeast and for the most part, and also flies and mice, you get 20, 30, 40% extension, but you don't get anything that's in the 10-fold magnitude that you can get by combining different mutations in worms. What is it about worms that make them so amenable to that extreme longevity? Is there something to be learned there from that?
Well, for sure. I think, the worm, so you can't understand aging and longevity without understanding evolution, nor can you understand it without understanding ecology. And the ecology of the worm is that it lives, on rotting fruit, that grapes that fall down in fall, and then they have to overwinter and survive. We're talking about survival in Germany over the winter. So they have to have robust mechanisms that enable them to survive over winter under conditions where it's not favorable to reproduce. There's not much food around. And so I think the reason for this, and it's no surprise that the original mutations that were identified were in this Dhfr pathway. So they have this mechanism that allows them to outlast winter. And it can be deployed in the adult to double or triple the lifespan. So they have enormous plasticity that has been selected in evolution as part of their ecology.
Yeah, I just had to survive Germany for a month, and that was a challenge. I had to look for...
Dhfr is a German word. That's where Dhfr means to, endure. So, there's something to it.
This has been, I think, another surprise in the aging field over the years. It's relatively easy to extend the lifespan of an organism. I think a lot of people in 1990, when I was getting in the field, were thinking it's going to be extremely hard. That was the early days where the worm genes had been identified, but not really widely accepted yet. And people were still saying it was going to be really, really hard to extend lifespan by modifying one gene. And yet it proves to be relatively easy. And I think evolution is a good way of thinking about that too, because evolution cares about fitness and not longevity. So when you get these longevity effects in worms that are so extreme, are they adversely affecting fitness, or are they neutral to fitness? And by that, I mean reproduction.
Yeah, I think that's an important question. And first of all, I agree with you that, the field, we take it for granted now that there's evolutionary conservation of these signaling pathways. They may have different magnitudes in different organisms, but, back in the day when we were starting, you and I think most people thought single gene mutants, that must be just, worms or yeast, not higher organisms. And, I think, the magnitude of the effects in worms, as I said, is probably linked to their ecology. And then the question of fitness depends on what you're measuring under what condition. So if you take, there's some elegant studies by Gordon Lithgow where he had a competition between wild type and DAF2 mutants and to see which one would prevail when you gave them plenty of food. And of course, wild type can outcompete the DAF2 mutants because they're a little bit slower to reach maturity. Actually, their broods are fairly comparable to wild type, but slightly less. But those slight differences on an evolutionary scale mean a lot. On the other hand, if worms had to, live on an asteroid or survive, nuclear winter, I think the DAF2 would probably do better than wild type because it has invoked such a strong, and robust stress programs. It's sort of overboards compared to wild type. So I think it depends on the selective environment.
Yeah, I agree. I think it's an interesting challenge to think about, though, because, we're thinking about modifying genetic pathways either directly or through small molecules in humans to affect longevity. And, the long-term side effects of that are still, so you need to think about fitness at some level. Now, it may be possible to avoid the fitness deficits by modifying things in an adult rather than in a developing person. And so there may be ways around the problem, but it's certainly something we have to keep our eyes on, I think.
Absolutely. I think, I would never say go out there and restrict your folate and, expect that could, we have to understand these regimens and what impact they may have on other systems of the body. And, they probably will have fitness costs if we don't look at this correctly. I think one way around it, as you said, is, to maybe some of these pharmacologic or metabolic interventions could happen post-reproductively and, or, in mid-age. And the other thing is to think about regimens that aren't constantly, where we're not constantly giving these agents, but giving them in bouts so that there's a period of intervention and then recovery. And I think it's actually that cycle of intervention and recovery that will give the most benefit. That's my hunch.
So I want to bring in Dong Lu, a graduate student in the lab, and she's going to ask some of the questions from the audience. Before I let her start, though, I just want to tell Joe we might run just a few minutes over because somebody on this show was asking too many questions. Dong Lu, you want to, what's the audience think?
Thank you, Brian, and thank you very much, Prof Antebi, for the illuminating talk. Now I'm going to start with some of the questions from the audience. It seems like many of our audience are interested in the translational aspect. So we have a question from David. He was asking whether it has been verified in humans that there is a downregulation of folate and methionine cycles in multiple longevity pathways. If so, what are some practical strategies that we can do to restore these cycles?
So in answer to the question, to my knowledge, no one has really looked at that. And I think that's largely because the idea that this could be a convergent mechanism is really new. But I think it certainly should be. I think it'd be very interesting to just probe the data from centenarians and their children to see whether they have polymorphisms in these pathways or whether these metabolites are altered. But for sure, that would be really interesting.
Yeah, that's a very interesting answer. Thank you, Prof Antebi. So if we continue about this topic, what are some first clinical symptoms one may experience from downregulation of folate and methionine cycles in the multiple longevity pathways?
Is that you're talking about in worms or in humans? I wasn't
in human beings. Of course, humans. We can also talk about worms.
So I don't know that it's really been systematically looked at. What I can say is that there are people who are looking at diet restriction and low amino acid diets and their impact on various types of surgery and also cancer. And so in some of those cases, those type of amino acid lowering diets can help synergize with chemotherapy. So Walter Longo has been looking at this to help arrest or kill off cancers. And when it comes to surgeries, I know that there are people are looking at ischemia reperfusion and looking at, for example, when you have heart surgery, you get some type of ischemic injury in the kidney. And people have found that diet restriction has actually a benefit outcome. It prevents that type of damage when you have a diet restriction preceding the surgery. Whether that's true for methionine restriction or folate, dihydrofolate reductase inhibition, I think that remains to be seen. But I think it's a really interesting question.
I think it's a really important question too, because we have one camp out there saying, low amino acid, low animal protein diets are good for you, and fasting, and another group saying high protein, high fat, low carb diets are good for you. And we need to understand that better, because we have people trying to do both. And I'm not sure if one size fits all, but it's an interesting question in the field.
Yeah, there's some people here at the clinics that are trying to address that now. So it's really very topical. I would add to that, people tend to focus on the fasting side or the restriction side, but I think equally important here is the refeeding. Fasting and refeeding bouts, I think, are important for rejuvenation. We're not like worms, which are mostly in their adult stage post-mitotic. We have stem cells that perhaps they're quiescent states. They are very, it's important to keep the quiescent so that they don't accrue damage, but then when they need to be activated through refeeding or nutrient sensing, they have to do their job and replenish the tissue.
And I think that another important thing out of that may be that it restores sort of the dynamic regulation of the TOR pathway. With aging, TOR tends to get out of control, and periods of fasting may sort of bring it back under the normal regulatory components that you want it functioning.
Yeah, no, I agree 100% on that. We're actually have. We're looking very intensely also in C. elegans in the diapause states, because they're really incredible, where they can survive very long under fasting. And then when you refeed, they rejuvenate a lot of their tissues. And so we think this could be a very nice model at an organismal level, and we might be able to understand better some of the molecules involved. So,
okay, we have more questions. I'll try to be quiet.
Oh, sorry.
No, it's me.
Yeah, we have a lot of questions. So actually, the audience are interested to know about the folate supplementation before any early pregnancy. Would that have an impact on the regulation of folate cycles or even lifespan in the mothers or their babies?
That's a great question. And, folate is often supplemented. And the question is it doing harm or good? And I think for the most part, in a developing fetus, it's probably doing good, because there's a lot of, cell division and DNA synthesis. And in humans, they have that as well. I think the good news is that, when we supplement folate to the worm, we didn't actually see a clear benefit for lifespan, but it didn't cause any harm either. And in fact, what I didn't show you was that it looks like it feeds back on the folate cycle pathway in high levels. It actually reduces the levels of some of those other intermediates in the pathway. So there's a kind of feedback regulation, at least in C. elegans. And so I don't think it's doing harm, and maybe elevated levels of folate actually could do some good. And also in support of that, I didn't really point it out, but when we give worms the folate acid, it didn't extend the lifespan, but it did ameliorate this proteotoxic model. So maybe it could even, who knows, maybe it could be used to treat neurodegenerative diseases or other proteotoxic diseases. But I think we really, going from worm to human is a long way, but I think we have the tools to do it now.
So I'm kind of going to the next topic, but it's also about the translational aspect. So many audience are interested in DHFR inhibition. Colin is asking, does DHFR inhibition increase hydrogen sulfide signaling?
Great question. We don't know the answer to that. My guess is it probably does, because it causes elevation of homocysteine, and that should feed into the transsulfuration pathways, but we haven't actually measured it, and it's certainly something we'd love to do.
Colin, if you ask questions, we're going to have to get you on the show, so be careful.
And we also have a question from Prof Barry Halliwell. Methotrexate is often regarded as a terrible drug, but at low dose is good for RA and maybe good for health. Why is that so?
Yeah, I think so. The folate cycle affects many different aspects of metabolism. It affects methionine pools. It affects the production of purines and pyrimidines and fat. And I think at high levels, it disrupts DNA synthesis and can cause DNA damage and ultimately apoptosis. So at that level, it's toxic and dangerous. It is in fact used to kill cancers. But at lower levels, I suspect that what it's doing is it's impacting amino acid pools or it has a more subtle effect, which is invoking a stress response that's actually protective. And so the dosing here would probably be pretty important. And I should mention there are other DHFR inhibitors that could perhaps work better. So I think that has to be really tested.
There are many hallmarks of aging in human beings and mammalian models. What are the ones commonly used in C. elegans besides lifespan? And how would those differ from other species, for example, mice?
Yeah, well, many of the hallmarks of aging, the ones that have been described in this very famous articles, they're mostly molecular hallmarks, and I think they span taxa. So you have in worms, you have failure of protein homeostasis with age, as you do with mammals. You have DNA damage accumulation. You even have metabolic dysregulation. Nutrient sensing pathways go awry. And there are epigenetic factors as well. I think what perhaps might be different is that because C. elegans is mostly postmitotic after it's reached adulthood, except for the germline, you're not going to see a whole lot of stem cell aging. You're not going to see, I don't know that there's a really good model of cellular senescence. Which is a very, important aspect of mammalian aging, we think. And although worms do have a type of inflammatory increase with age, similar to higher animals, the inflammatory pathways are somewhat diverged. For example, they don't have an NF-kappa B, they don't have C-gas sting, they don't have IL-1 or IL-6, the traditional, inflammatory pathways that people often invoke. That being said, I think we can understand a lot about what's happening, with innate immunity. Worms are really a great model for that. And also for physiology, I think, worms undergo sarcopenia, just like most species. Their muscles deteriorate with age. Their nervous system, the ability of the nervous system to function also deteriorates with age. They undergo, sometimes necroptosis. And so I think there are a lot of parallels, mitochondrial decline with age. So I think there's still a lot to learn. I think where the main point where I think they're not as strong is in the post-developmental cell biology, cell senescence and stem cell.
So now I'm kind of going to the next topic. Q-Chin was asking, could late life supplementation with one carbon metabolite potentially still promote longevity?
Possibly, yeah. I think that's certainly, if we, late life supplementation with rapamycin certainly has benefits. I don't think we can rule out that, manipulating the folate cycle could also have benefits. We haven't tested that yet. What I do know is that in our experiments where we supplemented with 5-methyl-THF, those were from adult on. But we didn't look at late life supplementation. I would add that, I think we do have to be very careful about understanding the aging trajectory, because it's not, I think there's this period during, up to midlife where you have different processes going on than late life, and where an intervention might be beneficial, say diet restriction in between the age of 40 and 70 may be beneficial. Maybe from 80 to 100, it's not. And I think this may be also true. People often they're not treated with anti-cancer drugs late in life. So I think we have to understand the aging trajectory of these interventions, and also think about personalized medicine if we're going to have successful and healthy aging.
Okay, I've already drug this on 5 minutes late, and I know you're standing up for this whole thing. So we're aging people.
Standing is good for your health.
So thanks a lot for joining the show, and please use the chat and panelists and all attendees option to add comments about the show. Also, we have news on courses and clinical trials in the credits, so look for that. Next week, we'll have Evelyn Bischoff, who's a professor at Shanghai University, not only a scientist, but a clinician doing longevity interventions in people. So it'll be exciting to talk to her. And with that, I'll leave you with our final clip, which is about some recent world records in track and field. Thanks a lot.
I like the idea of being 100 and doing the 100 meters. Somehow that was a little bit of a challenge. It sounded good. And so I decided that I would try it.
I was over at her house, just helping her do things. And then I was about to leave, and she goes, "Oh, one more thing." And I said, "What's that?" She says, "Time me in the 100. I go out to the street, and she takes off running, and I was surprised that, I wasn't expecting actual running. I'm not sure what I was expecting.
Julia would break the world record in the 100-meter dash for participants over the age of 100.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.